All right, let's dive right in. Today we're talking about the genetics of bipolar disorder, and we're going to break it down with a guide that's built specifically for the kinds of questions you face every day as a psychiatry resident. You've seen this in your own work. You have a patient with bipolar disorder, you start taking a family history, and you begin to see a pattern. It just seems to run in families. It's an observation we've all made. But what's the real evidence behind it? How do we go from that clinical hunch to the hard science? What's the data really tell us? Let's start with a number that really hits home. 10.1. That's how many times more likely a first-degree relative, we're talking parents, siblings, children, is to develop bipolar disorder. 10 times. That isn't just a small blip on the radar. It's a massive flashing signal telling us that something very significant is being passed down through generations. And that something, as we're about to see, is overwhelmingly genetic. So here's our game plan. First, we'll lock down that familial link with solid data. Then, we'll join the hunt for the actual genes, which leads us to the genome-wide revolution that really changed everything. After that, we'll see what modern genetics can tell us about clinical features, and then, the most important part, what does all this mean for you in your day-to-day practice? Okay, so, Section 1. Let's tackle that foundational idea. We all kind of feel that it's a familial condition, but let's look at the hard evidence that nails it down. And there's no better evidence than twin studies. I mean, look at this. For identical twins who share literally 100% of their DNA, if one twin has bipolar disorder, there's a 49% chance the other one will too. But now look at fraternal twins, who only share about half their DNA, like any other sibling. That number plummets to just under 12%. That huge gap right there, that's our smoking gun for a powerful genetic link. And the big analyses of all these studies really spell it out. They say, sure, the environment has to play some role. If it didn't, that identical twin number would be 100%, right? But the overwhelming reason we see this illness cluster in families, it's the genes. Okay, so we know it's genetic, but that just opens up a much tougher question. Which genes? And this question kicked off what was basically a decades-long detective story to find the specific culprits. The first attempts, they made sense. Back in the 80s, they tried linkage studies. They'd find these huge families with lots of members with the disorder and try to find a genetic marker that always traveled with the illness. The problem? The results were all over the map, super inconsistent. So then they tried candidate gene studies, where they'd focus on the usual suspects, genes for serotonin, dopamine, that kind of thing. And they'd get these promising results, but it often turned out to be something called the winner's curse, basically a statistical fluke that couldn't be repeated. It was becoming clear there was no single bipolar gene to find. The best way to think about it is this. Those early methods were like trying to find one specific house in a huge city, but you have no map. You might get lucky, you might stumble into the right neighborhood, but you're just wandering. The science needed a completely new approach, something like a satellite view. And that satellite view is exactly what we got with the genome-wide revolution. This is where modern tech, specifically what we call genome-wide association studies, or GWAS, just blew the doors off the whole field.
A GWAS lets you scan the entire genomes of thousands, even hundreds of thousands of people at once. And this timeline just shows you the insane result. In 2007, the first study found one genetic location, or locus, just one. By 2019, we were at 30. By 2021, 64. And in the most recent paper from 2023, that number exploded to 298. That's the power of having gigantic sample sizes.
Now hold on, before we go further, there's a key term we got to understand. SNP heritability. It's a bit technical, but the idea is simple. Twin studies tell us about the total genetic risk. SNP heritability, on the other hand, tells us how much of that risk we can actually account for using the common genetic variations, the SNPs, that these huge GWAS studies measure. So with 298 loci found, how much of the story can we explain? Well, right now, the estimate is that all these common variants explain about 22% of the heritability. This leads to this idea of missing heritability. It means that even with all these amazing discoveries, we still can't explain the whole picture we see in twin studies. That missing chunk is probably a mix of rare genetic variants, really complex gene-on-gene interactions, stuff we're only just starting to figure out how to find. So we've got hundreds of these genetic signals. Awesome. But what do they actually tell us about what bipolar disorder looks like in the clinic? How do they help us make sense of its complexity?
Well, one of the coolest things to come out of GWAS is seeing the genetic overlap between disorders, and there's a really clear pattern. The genetic risk for bipolar 1, it actually correlates more strongly with schizophrenia. But the genetics of bipolar 2, that lines up much more closely with major depressive disorder. This is giving us a real biological basis for the clinical differences you see and think about every day.
So why does that overlap happen? The key idea here is a concept called pleiotropy. It just means that a single gene can affect multiple, seemingly unrelated traits. A classic example is a gene called CACNA1C. It's involved in calcium channels in the brain, and variants in this one gene are strongly linked to the risk for both bipolar disorder and schizophrenia. This is exactly how we can start to explain that clinical and genetic overlap we were just talking about.
Which brings us to the big finale, the real bottom line. How do you take all this knowledge and actually use it in your practice? What can you do with this information for your patients tomorrow? Okay, this is absolutely crucial. We have to manage expectations. Right now, today, what is our best tool for assessing genetic risk in a patient? It's still a really well-taken family history, the clinical gold standard. You've probably heard about polygenic risk scores, or PGS, which add up the risk from thousands of gene variants. They are an amazing research tool, but for predicting if any single patient will get sick, right now their predictive power is actually lower than a detailed family history. They are not ready for prime time in the clinic. So since family history is our best tool, we have to be good at it. A quick, hey, anyone in your family have mental health issues? Just doesn't cut it. Research shows those histories are often wrong. You need a system. Go through each first-degree relative one by one. Mother, father, every sibling. Ask about specific diagnoses, psychiatric hospitalizations, specific treatments like lithium or ECT, and you have to ask about suicide. And whenever you can, get a family member's permission to talk to another relative to confirm the details. That's how you get a history you can actually rely on. But there is one place where genetics is absolutely in the clinic right now, and that's pharmacogenetics. We can already do things like genotype for PSYWIP enzymes, like 2D6 and C19, to figure out if someone is a poor or an ultra-rapid metabolizer of certain antidepressants and antipsychotics. And even more critically, we can test for specific HLA variants to predict the risk of a catastrophic rash like Stevens-Johnson syndrome with drugs like carbamazepine or lamotrigine. This is personalized medicine, and it's happening today. So let's end with a look to the future. I want you to picture your patient, 19 years old, walks in with their first major depressive episode. The million-dollar question is this unipolar depression or is this the start of bipolar disorder? Now, fast forward 10 years. Imagine you have a powerful, validated genetic risk score in your toolkit. How does that change your assessment? How does it change how you counsel that patient and their family? How does it change your very first treatment choice? That is the future that you are going to be a part of. Thanks for joining this explainer.